All right, welcome to another screencast. This is about scientific investigations, just good for you to have as an awareness that we have other types of investigations beyond the ones that you've most likely done throughout your high school career. So, this dabbles in with just the basics of a scientific investigation, but then it also talks about these other ones, which are extremely important for apes this year. All right, so our goal from these investigations is that in the end that we can further understand these natural systems. The wonders of science is that there's no confetti cannons when we find the cure for cancer or discover a new vaccine. It's constant questions and desire for understanding and knowledge. It's fast. I think it's fantastic. I absolutely love it. So more on that later. But all of it is gathering evidence, evidence, evidence. And we've got two different types of evidence that we're going to specifically focus on. Most importantly in apes, qualitative and quantitative. I know most time in your previous classes, especially chemistry, it is almost always quantitative data. Now we're going to bring in qualitative. So qualitative is non-numerical quantities, color, smell, in apes especially behavior. And then we have our quantitative data as well, which is our numerical values. All right. And then anytime we do a scientific investigation, we always involve variables. Of course, you've seen these before. Independent variables, those are the ones that we are intentionally changing by the investigator. Dependent variable, that's going to change in response. And then the controlled variable, that's one that we're going to keep constant so that we know what the baseline is, and we can look for change in the dependent variable based on how we changed our independent variable. Most important in apes, and I don't think this is something that has been talked about much in your other courses, is something called a control group or a sample group. This is going to be a population that is left untreated or unexposed to the change. And it's always used for a purpose of comparison. It's extremely important when we're looking at environmental changes because we need to know how would that species continue on if it was left alone. Maybe they all just died out and it had nothing to do with our independent variable change that we exposed it to. So these are used for the purpose of comparison to determine the effect of the independent variable. Very important when we get into apes this year. A great example here is if I want to use pesticides to look at plant growth. I'm going to take one experimental group and expose it to pesticides, but I'm going to take a control group and not expose it to pesticides. So then I can see what the differences are between the two. Maybe there was something in the environment that killed these plants and it had nothing to do with the pesticides. We would never know that unless we had that control group. All right, so as I mentioned, beyond what you may have learned previously in your science courses, there are different types of scientific investigations that we are going to rely on heavily this year. The first is something called a descriptive investigation. This actually leads to very little conclusion at all. You don't have a hypothesis. It's really just describing and quantifying what is happening in a system. This could be a coral reef, it could be an ecosystem, it could be a garden, it could be a riverbed, but we're just describing it. That's all we're doing. We're not really analyzing the effect of something. We're just trying to get a update of the status of everything. How many species do we have here? What are our quantitative and our qualitative variables that we notice here? Comparative is when we're going to be collecting data on a system or population that's under different conditions. This is very similar to before with the plants. I give one of it pesticides, one of it not pesticides. I'm going to compare the results of each of them. This is where we're going to have a traditional setup with a hypothesis and our procedure and looking to analyze the results of the two of them. And then the one you're probably most familiar with is the experimental. This is where a variable is changed while other variables are kept constant to determine the relationship between two variables. We're going to look at all three of them. However, I want to just remind you that there's more types of investigations than just the experimental, which I know you did a ton of in regular chemistry. So we're going to move on beyond that. Okay, let's look at this descriptive investigation. This involves making observations, notes, measurements, looking at a natural system and trying to understand what is happening. It is a lot of data using some very prescribed methodology. There it is. As I mentioned before, there's no hypothesis, no prediction. We are really just looking to see what is going on here. We will reach a conclusion, but it does not involve an independent, dependent, or controlled variables. We're not making a conclusion based on a reactionary or reaction to something that may have happened. So one example, move my face here a little bit. So an example of this is we can look at the size and distribution of bird colonies on an island. The salinity of the water is in different parts of the marsh, and it's recorded along with this fish species that are in those different areas as well. So we're just doing lots of recording of the fish, of the birds, of the size, of all of those things. Lots and lots of recording. This is different from a comparative investigation. This is where we are collecting data on a variable, whether that is population size, amount of runoff, CO2, under these different conditions. One area is going to have one condition, the other area is going to have a different condition, and we're going to make a comparison based on that. It does involve a hypothesis and an independent variable and a dependent variable. And most of the time, we have to have a control variable in this situation because we have to know how this area, whether it's population, runoff, how it would be affected if it was just let to be without anything interacting or interfering with it. This is almost always done in a natural setting, not a lab setting. So here's an example. We're looking at the volume of runoff in two streams. One stream's in a very forested area, the other one's in a very clear-cut area. So our controlled variable would be a similar land area with a similar slope and the similar rainfall amounts. Another example could be fertilizer given to one plant, but not to another. After a period of time, we're going to compare the growth of those two plants. So we're comparing one that received the treatment and one that did not. And then finally, we have the experimental investigation. This is one that, like I said, you have done most frequently in your academic career. So for this one, we have our independent variables incrementally changed. So it could say, maybe we're going to use light. All right, we're going to expose this plant to harsh light. We're going to expose this plant to 50% light. We're going to expose this plant to 10% light. We did a lot of this in chemistry with dilutions and concentration. So what happens if we use 1 molar or 2 molar or 3 molar? So we're taking the same variable and we're incrementally changing it by the investigator. So we can see a very clear relationship between the independent and the dependent variable. This involves a hypothesis, all the variables and especially a controlled variable, even though most of the time we don't really notice our control variable, especially in chemistry, was often just water. Often this is done in a laboratory environment because we need to keep our control variables as constant as possible. We need to make sure the only thing that is changed is our independent variable. An example of this could be the brightness of light of photosynthesis. It's investigated by exposing plants to various levels of light. That's kind of what I was talking about before. Control variables would be same plant species, same size of plant, same amount of water given to the plant. So we often have so many controlled variables, which is why this happens in a lab, because you really can't control for all these variables if this is happening out in nature. All right, that is all three of them. That's a wrap. I look forward to getting in and actually doing these with you. Fantastic.